All right, guys, I'm going to try this again. This is a third attempt at recording this. I don't know why we're having such problems, but let's start with a summary from the last time that we noted. What are three types of transport across cell membranes and the three types that we've talked about? We talked about passive transport, which is diffusion, just going from high concentration to low concentration. We've talked about facilitated diffusion, in which we've got something like a polar molecule or a charged ion that's going to have a hard time making its way through the hydrophobic region of the lipid bilayer. So it's going to need help from a protein in order to make its way through. That's facilitated diffusion from high to low. And then we're going to talk about active transport here today, in which we've got going from low to high concentration, actually. Which ones involve the invest of energy? Well, the active transport does, because it's going from low to high energy, and I'm going to use an analogy to try to explain that today. And then what are three types of solutions that describe concentrations of those solutions? We've got hypertonic, in which we've got a lot of solute, hypotonic, in which there's less solute, and isotonic, in which there's a relatively equal amount of solute, in which there will be no net diffusion, or we could say it's reached dynamic equilibrium. Okay. We also used this example last time. I talked about a weird example in college, which I had somebody come sit right next to me on a bus when I was sitting completely alone. Things usually like to be spread out, right? And obviously this is a limited analogy of what I'm talking about with particles. Particles will naturally want to do this. They'll want to go from high concentrations to low concentrations. Things generally don't like to be packed in high concentrations. That causes high amounts of energy. A really good example of how this is going to work, and I'll use this example several times as we're starting to talk about energy, is it's almost like the ball on top of a hill. If you give this a little push, it's naturally going to roll down here, down to the bottom, and it'll be lower in energy down here. It'll be a lower energy state than it was up here, less potential energy, right? And this is something that's just going to happen. This is like diffusion, going from higher energy to lower potential energy. In order to get that back up to higher potential energy, you'd have to invest energy. You would have to push the ball up the hill. This one just rolls. This one you'd have to push the ball up the hill and expend some energy. It's the same thing when we look at things like diffusion. In order to get things going from against their concentration gradient to go against the concentration to push up against the energy, you're going to have to, that requires energy in order to do that. So active transport uses energy to move solutes against their concentration gradients, right? So we're going from low concentration in active transport to high concentration of a solute. That's going to require energy, usually in the form of ATP, adenosine triphosphate. How does it do that exactly? We're going to look at an example here called the sodium-potassium pump, which is a really good example of active transport, moving things against their concentration gradients. Here's a summary of the three types of transport that we've talked about thus far. Now, we've got diffusion, in which we've got something like a small nonpolar molecule, which can make its way through this hydrophobic region in the lipid bilayer from high concentration to low. Right? And then we've got something like maybe like water or a charged ion down here, a small particle that's going to make its way through a channel protein through facilitated diffusion. It needs help. So it's going to go from high to low concentration. That's what these two things have in common. The difference with this is that it needs help from an integral protein within the lipid bilayer. Then down here we've got something like a carrier protein, and notice the difference. We've got a low concentration moving to a high concentration. It's going against its concentration gradient, and that's going to require ATP in order to do that. An example of active transport, like this example down here, is the sodium-potassium pump. This is actually how we send electrical signals in things like nerve cells. So up here, notice what we've got is an integral protein embedded within the lipid bilayer. Here we've got the cytoplasm and the extracellular fluid. Out here we've got a high concentration of sodium. Down here we've got a low concentration of sodium. The sodium is going to embed its way into this protein, and by doing that, that sodium binds. That's going to stimulate the phosphorylation by ATP. So ATP comes in, it's going to bind a phosphate group to this protein here, and now when it does that, it causes a conformational or a shape change in the protein, in that integral protein. It opens up to the outside, and now the sodium gets released. Right now down here, the potassium is free to go ahead and bind to that protein, so potassium is going to do that, and it's going to make its way inside of the cell, where it will go from low concentration to high concentration. So the sodium-potassium pump is a major consumer of energy. It's one of the main reasons why humans have to eat so much food relative to our body weight, is because we expend so much energy in the sodium-potassium pump in order to send electrical signals. We have such big brains that we need a lot of energy to supply all these signals that are sent. So what happens here is the sodiums will build up a positive charge here on the outside, which makes the inside of the cells relatively negative compared to the outside. because there's more positive out here than there is in here. That will build up a membrane potential. It will build up a voltage gradient in which now an electrical signal could shoot along this accent and can send a signal. Right, so the difference in overall charge between the inside and the outside of that cell is called a membrane potential, in which the sodium, which Drake is doing over here for us, is sending sodium across over on this side, and then we've got potassium pumping across this side. So that difference in charge causes these sodiums also to be attracted to the cell, sort of like sodium will be attracted to that kind of negative inside of the cell, so they will gather up and that will store a membrane potential. It will be like a charge. The last type of transport we're going to talk about is bulk transport, right? So this is transport 4 and 5. So far we've got passive diffusion, we've got facilitated diffusion, we've got active transport, now we've got endo and exocytosis. Exocytosis and bulk transport is for big molecules. So what is going to happen with exocytosis is vesicles from the cell's interior, from already inside of the cell, maybe something like a protein, is going to bind with a vesicle. It's going to be packaged up, which is made out of a lipid. It will then bind and fuse with the plasma membrane, in which those secreted proteins will be sent outside of the cell. Out, exocytosis. That might happen in something like cell communication, which this is a signal that could be sent. The opposite of that process is called endocytosis, in which particles are brought in. So this could be like a food particle right here. The cell is going to form a new vesicle, maybe due to some sort of glycoprotein, a receptor that will only bind to this particular type of particle. It will then signal a pinch to happen of the plasma membrane, in which those particles are taken in, and once those particles are taken in, they might be digested. This is how macromolecules are brought into the cell through digestion. Right, so an example here could be maybe a food particle that's on the outside of a cell. The plasma membrane is going to form a food vacuole around on the outside. Lysosomes would then bind to the outside of that, and they would digest that food particle. Because there are receptors around here on the outside, the lysosomes can bind, they'll start digesting the food particle. That is endocytosis, bringing something into the cell. And that's all the types of transport that we need to know, guys. So that quick video hopefully helps you fill out those notes. If you have any questions, please be sure to ask me.